This video we are talking about UTP. UTP means what? Uridine triphosphate. So what are the components of uridine triphosphate? So there is a ribose sugar. So this is called a uracil. The uracil is linked to the ribose sugar by N-glycosidic bond. This bond is called N-glycosidic bond. And on the fifth position of the carbon atom is associated with the phosphate group. There are three phosphate groups, 1, 2, 3. So it's called uridine monophosphate, uridine diphosphate, and uridine triphosphate. So these are linked with the ester bond. So this is the basic structure of uridine triphosphate. It's a pyrimidine nucleotide. Why? Because the uracil is a pyrimidine consisting of the uracil linked to the one carbon of the ribose sugar as told esterified with the triphosphoric acid at the fifth position. The main role is as a substrate for the synthesis of RNA during transcription. So this is by which the RNA is produced during the transcription. Transcription means what? Formation of RNA. So what are the other roles of UTP? It has the role of source of energy just like ATP. When UTP activates, UDP substrate complex is usually formed and inorganic phosphate is released. For example, in the synthesis of glycogen or glycogenesis, the glucose has to be added to the glycogen chain. So the glucose cannot be directly added to the glycogen chain. Before that, the glucose has to link with the UDP. So when the UTP plus glucose results in UDP substrate. Substrate here means what? Glucose. UDP substrate is formed and an inorganic phosphate is released. So this UDP glucose enters the synthesis of glycogen. UTP is used in the metabolism of galactose also, where the activated form UDP galactose is converted to UDP glucose. So NAD has four components, namely a nicotinamide. This is a nicotinamide. An adenine group. This is adenine group. Two pentose sugar, one here and one here. Two phosphate groups are there. One is here, the other is here. Two phosphate group, and it acts as a coenzyme. It plays an important role in the oxidation of glucose and fatty acid. These are linked to this is N-glycosidic bond. This is also N-glycosidic bond. So this is a ester bond. There are two ester bonds are there. During the oxidation of these molecules, the electrons are transferred to oxygen through a special carriers like NAD and FAD. From the NADH or FADH, the electrons are transferred through a special carriers like a coenzyme Q, cytochrome C, and finally to the oxygen molecule and forms what? Water. So it has an important NADH and FADH has an important role. So they act as an electron carrier in the electron transport chain. Thus NAD is a major electron carrier and acceptor in the electron transport system. Between NAD and NADH is here. There will be an additional H group in the NADH. And NADP is what? NADP has the same structure as NAD, but there will be a phosphate group in the second carbon atom. This portion is the first carbon atom, is the second carbon atom. There will be a phosphate group attached here. So then it becomes what? NADP. The coenzyme, this is also called coenzyme. The coenzyme nicotinamide dinucleotide phosphate is another coenzyme related to NAD. It also functions as electron acceptor. When it accepts electron, it is reduced to NADPH. And both of these nicotinamide ring is the most active part. It accepts hydrogen ion and two electrons. Both involve the dehydrogenation reactions such as glycolysis, Krebs cycle, beta oxidation, DNA splicing, etc. So these are the roles of what NAD. It involved in glycolysis, Krebs cycle, beta oxidation of fatty acid, DNA splicing, etc. When NAD accepts electron, it is reduced to NADH.